This is a quick demo on how the Avardis platform at Strand Life Sciences can be used to import and analyze flow cytometry data. Flow cytometry data is usually captured in FCS files, and in Avardis we have here a short script about a couple of hundred lines that we have written that will automatically parse an FCS file and do further analysis on it. In this case, the FCS file of interest actually comes from an HIV patient, and the goal is to count the number of CD4 and CD8 cells in this patient. So all of this will be done automatically by this script. So let's go ahead and run it. It asks us to choose a file, and we can choose one or more FCS files. So let's choose a few of these and then run it. It asks where it should store the results of the counts of the CD4 and CD8 counts, and it asks us to choose a beat count for normalization, and we hit OK, and in a moment it will import in each of these files one by one. And you'll also notice that for each file it does some automatic processing. So it looks at these two channels, for instance, and identifies this cluster of points, which comprises the CD3, CD4 and CD8 cells all together, and the beads are separated out here. So the import script automatically segments these populations and identifies the beads, normalizes the beads based on the numbers that we provided earlier, and then takes this population of CD3, CD4 and CD8 all mixed together, and based on these two channels again separates them out and counts each of those individually. And the individual counts are presented here. So we know the CD4 count now is 383. After normalization, the CD8 count is 1294, and so on. So, and as you can see, each of the plots is appropriately colored so that what the algorithm did by way of gating and clustering, automatic gating and clustering, is directly visible and verifiable. So that was a brief demo of how the Avardis platform at Strand Life Sciences can be used to import and analyze flow cytometry data.